Okay, so today we are going to continue our chapter on DNA, and today we're going to concentrate on S phase of interphase. In other words, we're going to talk about the synthesis phase on how DNA is replicated. All right, so here, and I know we've been kind of keeping consistent with colors, but here is the two strands of DNA, one here and one here, and these are our original strands of DNA. So no new DNA has been made yet. But what we're going to do first in order to replicate our DNA is we are going to make something called a replication fork. Now, how we do that is we are going to bring in this enzyme. This is going to be like our first main character we bring in. But it is called it is called helicase. And that is this enzyme right here. Okay, so what we're going to do is we are going to bring in helicase, and helicase creates this replication fork. And how helicase makes this replication fork is by breaking the hydrogen bonds in between our nitrogen bases right here. Okay, so helicase breaks those hydrogen bonds. Also, it unwinds our DNA. So DNA is always going to be in this helix form, in other words, like a spiral staircase. And what helicase is going to do is it's going to unravel that, okay? Now, if we were to, and think about this, guys, if we're unraveling it right here, right in this area, if we're unraveling it, this area over here is still going to be raveled up, even though it's not in the picture. But if we're unraveling it here, what's going on is originally, and I'm going to try to draw this to the best of my ability, originally this is still coiled over here, like this, okay? So notice how I did it. Notice my spacing in between, there's a lot of space. As we move closer to this area right over here, as helicase moves closer, what's going to happen, guys, is pretty much that DNA that helicase has not worked on yet, so this DNA right over here where I made my squiggly line, as we move closer to it, that DNA actually becomes like super coiled. So it's almost like if you have two pieces of string and you pull them like that, the raveling goes down and it gets, twisted up even more down this way because you're pulling them apart, okay? So as we move helicase closer to that area, what happens is right here, and notice how I have it, okay? It's going to go from looking like that to looking like this. All right, where it's raveled way too tight. So this guy right here, another enzyme called topoisomerase, his job is to make little adjustments to the DNA that's still bonded together to the other side so that it doesn't tear, pretty much. So topoisomerase and helicase, I don't want to say they work together, but they combat one another because helicase, it breaks those hydrogen bonds and unravels it, and topoisomerase, it kind of protects the DNA from the breaking that helicase has already done. So those are our first two characters I wanted to talk about, guys. The helicase, the topoisomerase, there's your notes on that. The other one is called SSBP, single-stranded binding protein. So that's these gray guys right here. Now, what we're going to do is, because we're trying to do the semi-conservative model, we're trying to make it so that each one of our new cells has one original strand of DNA and one new strand of DNA. So as helicase does its job and breaks all those hydrogen bonds, the DNA is going to split, make that replication fork. Problem is, as helicase makes its way in, well, the DNA on the other side of it, it could just bond back together. We don't want that, okay? Because there's going to be, a new strand being made. We want to make sure that new strand can be made. So as helicase does its job and we make that replication fork, which keeps moving, these single-stranded binding proteins, what they do is they pretty much take the DNA and hold it apart. So that way the DNA can't come and bond back together. In other words, those hydrogen bonds in this area right here, like between that one and that one, can't reform. Okay, so single-stranded binding protein keeps those DNA apart. So those are our first three characters. Now, if we look on the left side here, this is going to be a bacteria prokaryotic cell going through DNA replication. And notice that the DNA is circular, which it is. Bacterial cells have one DNA and it's circular. Our DNA, we have many different strands, many different chromosomes, and ours are linear. But if you notice here, we have a replication bubble for them because it's a circle. So what we're doing is we're trying to make a whole new DNA molecule there, okay? And it's going to be, in circular form at the end. For us, for our cells, we're going to have a bunch of bubbles, and it's kind of going to try to replicate our DNA all at once with a bunch of these different little bubbles. So we're going to have a lot of replication forks on ours, where the prokaryotic DNA, we're just going to have one, and we're going to synthesize our DNA both ways there. Okay. Now, as we start this process, we are going to start off with an enzyme called primase. And what we're going to do is we're going to use that primase, and we are going to make a RNA primer off of it. And that's what starts this whole process. So our whole process of copying DNA, which is really weird to say, but our whole process of copying DNA actually starts with RNA. Weird, right? You would think that, we're making DNA, should all be DNA. That just makes sense. So let me show you a picture here. Go back to this picture. We're going to start off with primase. And primase is going to bond to one of our sides here. All right. And I know we talked about our 3' and our 5' last time. We are going to continue to talk about that today, but I'm going to wait to bring that in a little bit. So primase is going to latch on to our old strand of DNA, and that is going to start our new strand of DNA being made. So this right here in red, that is our RNA primer being made. So primase attaches there, and wherever primase attaches, it's able to start to make our new strand, which is starting, like I said, in red here. And it's going to lay down the RNA primer. So what's going to happen is it's going to put this one down first right here. It's going to put this one down second, and then primase is going to move that way. We might have, 1, 2, maybe 3 more, and primase would end up over here in the end, okay? So that is our beginning of DNA replication. So we started off with helicase and topoisomerase. Helicase broke those hydrogen bonds. Topoisomerase made sure our DNA that was still bonded together didn't tear. Single-stranded binding proteins kept our 2 DNA molecules apart, so we're single-stranded. And then primase came in and laid our RNA primer. Now, after that RNA primer, and I'm going to come back to that RNA primer to talk about in a minute here, but after that RNA primer, we are going to bring in some DNA polymerase. If we wanted to be specific, it's DNA polymerase number 3. And it's going to attach to our primer at the end. So what's going to happen here is right after, let's say our RNA primer here, it should be a little bit bigger than this, a little bit longer, but let's say it's only 2 nucleotides long. Our next guy coming in right here is going to be our DNA polymerase. It's going to be abbreviated DNA Pol. DNA Pol. If you want to be real specific, it's number 3. And what it's going to do is it's going to start to lay down the tracks, lay down the new DNA nucleotides. So it's going to, like I said, attach to the RNA primer, and then it's going to work its way down. Now, as it works its way down, guys, it's going to move very, very, very quickly. It's going to go about 500 nucleotides per second in bacteria and 50 in ours, which is still pretty fast. And don't forget, I know in bacteria, you're like, oh wow, it's a lot faster in bacteria. But if we go back to this slide right here, remember, this is happening in a bunch of different areas on ours. So 50 nucleotides per second doesn't seem very fast, but there's more than one DNA polymerase working at one time because we have more than one replication fork. So it's actually doing, 50 per second, but there might be thousands of DNA polymerases working at the same time to accomplish this. Now, when we're talking about DNA, guys, and this is something we didn't learn in regular biology that we got to add on here. So we know that we have our phosphate attached to our 5-carbon sugar that's attached to our nitrogen base here. And that's our nucleotide. Now, DNA actually doesn't start off with just nucleotides that look like this. In reality, we have 1, 2, 3 phosphates here. So these are 3 phosphates. And we call this not yet a nucleotide, but we call it D-ATP. And this is it sounds like ATP, and it pretty much is. But it starts off as D-ATP. And then what we do is we want to turn it into a nucleotide. D-ATP, the A is the adenine, so this would be A. All right. We also have D-GTP, where it'd be the same thing, but instead of an A, there's a G. We also have D-TTP, where the A is a T. We also have D-CTP, okay? So we have all of them. And what happens here, guys, if I change my colors up here a little bit, we have 1, 2, I'm going to make the last phosphate a different color here, 3 phosphates. Attached to that, we have our 5-carbon sugar. I'll draw my oxygen in on this one. And then lastly, we have our nitrogen base. What happens here is the DATP turns into a nucleotide, and how we do that is by breaking this bond right here. So we are left with our 1 phosphate, just like we would have on a regular nucleotide. So this is DATP, and we're converting it into a nucleotide by breaking that bond. So what that does is it releases 2 phosphates that we can use for whatever else in our bodies or to make more nucleotides. But when DNA polymerase is going to attach our nucleotides to that new strand, what happens here, guys, is it originally is DATP, GDTP, GTTP, and GCTP. And what we do is before it attaches it, we get rid of those 2 phosphates, leaving just the nucleotide for DNA polymerase number 3 to attach. And this is just what we talked about here. Okay. Now, DATP, that's going to contain deoxyribose because it's a nucleotide for DNA. We would also have one for RNA as well, kind of the same concept, except it would have ribose. All right. Now, as we start to attach it, guys, we are going to make that brand new strand. And this is where our 5' and our 3' is going to come into play. DNA polymerase can only move 5' to 3' on our new DNA strand. So, let me repeat that. DNA polymerase can only move 5' to 3' on our new DNA strand. I'm not worried about the old DNA strand at all right now. I'm worried about my new one. So we got to learn which side is 5' and which side is 3' and pay attention to that to learn which way DNA polymerase is going to move. So there's going to be a leading strand and a lagging strand, okay? I'm going to show you guys, I'm going to go through all this, and I'm going to show you how to label your leading strand and your lagging strand. So instead of going over all the notes, guys, I'm going to draw you a nice picture. You can see there was lagging strand, leading strand, and then there's this other new term called Okazaki fragments. So let's draw everything out here. So I'm going to start with my old DNA here. there's one, and there's the other. We have our helicase here coming toward the middle, and it's breaking those hydrogen bonds. We also have topoisomerase over here. Making sure our DNA does not break. And then we have our single-stranded binding proteins here. Single-stranded binding proteins on both sides. Okay. Now, I'm going to say that, and I'm just making stuff up, guys. It doesn't matter which way you have this. I'm just going to say this side is for 3', this side is 5'. And that's all we need. We actually really only need one. I mean, I could even just say, hey guys, this one here, that's 3'. And you could label the rest. If that's 3', this side has to be 5'. If that's 3', this other side over here has to be 5'. And if that's 5', the other side over here has to be 3'. Okay? Now, we are going to start to make our new strand. But before I do that, I'm going to add some base pairs in here, because if I add base pairs, I think everything's going to make a little more sense for us. And then I'm going to do the same on the other side. So A would bond to T, T would bond to A. I did not like that T. Just didn't look right. T, A, A. We got a C, so that would be G. G would be C. G. A would go to T. And T would go to A. So you could see that they would fit together. The T would bond to the A would bond to the T, the A would bond to the, you know, you guys get where I'm going with that. Now, our next step is let's determine when we're making this new strand, which side is going to be 5' and which side is going to be 3'. We want to have a new strand right here and right here, okay? Where we're going to attach our nucleotides like that and like this, okay? I'm going to erase these in a second, just so we can add things, but I want to make sure you guys know what side is 5' and what side is 3'. If this is 3', that means this side would have to be 5'. That means this side over here would have to be 3'. If this is 5', this would be our 3', this would be our 5'. Okay, so that's there, okay, our 3', our 5' are all labeled. So you guys can see which one is going to go where. Let me erase them. And we are going to bring in our first characters for DNA replication of making the new strand. Now, I talked about DNA polymerase going 5' to 3'. So I'm going to do the 5' to 3' one first that is going toward the replication fork because helicase is moving this way. And you'll see why this is easier in a second to do it this way. So I'm going to start off with my primase. And we're going to start to add our primer, and I'm going to make it 3 long. In reality, it would be a little bigger than that, but that's okay. So 5' would be here, and our primase would be moving over, adding the nucleotides. Now, A usually bonds to T, but this right here is an RNA primer. RNA does not have T. And that's the only difference between RNA and DNA with respect to bonding. Another difference, RNA has ribose and DNA has deoxyribose, but A usually bonds to T, but there is no T, instead it's a U. And then everything else is the same. T still bonds to A, T still bonds to A. So there's our RNA primer. Now, we got to move 5' to 3'. RNA primer is done. The RNA primer allows our DNA polymerase number 3 to latch on. DNA polymerase number 3. And DNA polymerase number 3 is going to move this way. So I will lay my new track of DNA. We're going to have our G. We're going to have our C, G, T, and A. And if you notice, here, I didn't have to worry about using U's because we are back to DNA here. This is the blue is DNA, the red is RNA. Now, if you notice, we're moving 5' to 3'. And what's nice is we never really have to stop because we're just following helicase. Helicase, it's like a line, guys, okay? Think of you're in the cafeteria getting food. One person moves up and gets their food. You get to move a little bit closer to the front. You're just following helicase, just like DNA is here. All right, this right here, our new strand, this is called the leading strand. And the leading strand goes toward the replication fork. So toward replication fork. All right. Now, let's do our other strand of DNA. This one's the more difficult one. So, you would think we're going to go this way. But this is 3'. That means 5' would be over here. DNA polymerase can only move 5' to 3'. So we actually have to go this way. Get rid of that. Okay. So let's start going that way. So we will start off once again. Yeah, that was not good. Hold on. Let me make it a little further away. There we go. We will once again start with our RNA primer and our primase. A is going to go to U, T is going to go to A, G is going to go to C. That's 5'. We're then going to take our DNA polymerase in there. Okay. There we go. And we're going to keep moving this way if there is more of our DNA left over, because this over here is going to be our 3'. Okay, so we're moving that way. This here, that is called our lagging strand. And it moves away from the replication fork. Why we call it the lagging strand, guys, is because since it's moving away from our replication fork, we're going to go this way. Okay, we're going away from it, but helicase is still continuing to move this way. So we're going to have more DNA spit out this way, where we're going to have to form another one of these little guys. This right here, it's on the lagging strand, it's called an Okazaki fragment. And it's just a small section of DNA, all right? And we're going to have a bunch of these on the lagging strand because we're not following helicase anymore. We're actually moving away from helicase. So some DNA gets spit out. We make an Okazaki fragment, and then we're going to form another primer and make another Okazaki fragment and form another, or get another primer and form another one. And this process just keeps going over and over and over again, only on the lagging strand though. So the lagging strand has the Okazaki fragments, the leading strand does not. The leading strand is synthesized completely in whole. It's nice, one big long chain. The lagging strand, it makes a bunch of small fragments, and then we will later have to put all of those together. Okay. So after we have all these Okazaki fragments together, which let me go back here. You guys can see this one up here. I just want to show you. That's moving toward our replication fork. That again is the leading strand. The lagging strand would be at the bottom. I flipped it on my picture just to show you it could be either way. So after we form all of our Okazaki fragments, we have more than one DNA polymerase. We talked about number 3 already. But DNA polymerase number 1 then is going to get rid of the RNA, because we want this to be DNA after all, and it's going to replace it with DNA. Right? We are then going to have one more person come in, one more enzyme, and it's called DNA ligase, and it's going to seal all of our bonds that are in between Okazaki fragments. So the DNA ligase is going to connect all of our loose ends together. In other words, if we have a gap between our RNA primer and our DNA, it's going to seal that gap with a phosphodiester bond. It's also going to seal our gaps in between our Okazaki fragments. And here is the picture, guys, of the lagging strand. And you can see up here we have 2 Okazaki fragments. We have an RNA primer, DNA in baby blue. Again, another RNA primer and DNA in baby blue. And then what's going to happen here, as you can see at the bottom, ligase comes in. All right, we already had DNA polymerase number 1 come in, remove the RNA primer, and it's going to seal the gap in between them. And there is your final picture, guys, of everything, the leading strand. You can see it's one big, nice, harmonious picture of DNA, one complete strand. It's completely unbroken. Where the bottom one here, we got all these little Okazaki fragments on the lagging strand. Okay, so I think that's enough for you guys to chew on for today. That was a lot of information. Just please go over all of the different characters in that replication process. Know your topoisomerase, your helicase, your DNA polymerases, your primase, all of these different characters. And we'll talk more about it next time. Have a good day, guys.